Hi, everyone. Welcome. I see we're just at the bottom of the hour here, so I think we'll get started for today. Thank you for joining. My name is Christina Asa. I'm product manager here at Fujifilm VisualSonics, and I'm very pleased to be moderating our exciting webinar for today. Today, we'll be talking about non-invasive imaging of liver disease by MRI and photoacoustics, and we have two very distinguished speakers who contributed to this presentation, who are Dr. Chris Albanese and Dr. Olga Rodriguez, both from Georgetown University Medical Center. I'll start with just a few notes about our webinar for today. Hopefully, many of you have joined us in the past, so you know the process here, but we are recording our session for today, and we will make that recording available once our webinar has concluded to all those who have registered. Everyone's lines are muted for the duration of the webinar, but please use that Q&A button at the bottom of your Zoom panel at any point throughout the presentation to ask your questions. We will have a Q&A session at the end where we'll moderate all those questions to our speakers. We do expect our presentation to be about 45 minutes in duration, leaving us about 5 to 10 minutes for questions. Now to introduce our distinguished speakers for today. First, Dr. Chris Albanese is a professor of oncology and radiology and is the director of the Georgetown Lombardi Animal Models Shared Resource. As both the founder and director of the Georgetown Lombardi Preclinical Imaging Research Laboratory and the Center for Translational Imaging, Dr. Albanese has developed a comprehensive imaging program that spans basic, molecular, genetic, and translational research with multiple platforms, including MRI and ultrasound for in vivo modeling. As part of his basic sciences program, his laboratory and the PIRL-CTI are testing novel and emerging contrast agents. In addition, Dr. Albanese has over 30 years of experience in exploring the roles of oncogenes, tumor suppressors, and cell cycle regulatory proteins in the initiation and progression of cancer in vitro and in vivo. Dr. Albanese routinely provides guidance and performs imaging analysis on numerous preclinical models of human disease, from traumatic brain injury to cancers to heart disease. His overarching goal and the mission of the laboratory is to continue to enable the development of bedside to bench to bedside imaging programs using animal models as the nexus. Our second presenter or co-contributor is Dr. Olga Rodriguez, who is an associate professor in the Department of Oncology and the co-founder and co-director of both the Georgetown Lombardi Preclinical Imaging Research Laboratory and the Center for Translational Imaging. Dr. Rodriguez has an extensive background in clinical medicine, molecular biology, animal modeling, and preclinical imaging. She has over 15 years of experience in MRI and image post-processing and is instrumental in the translational research performed at Georgetown. Dr. Rodriguez is involved in several collaborative projects that include MRI, contrast agent development, and applications of MRI in various disease models. She's also a leader in enhancing equity, inclusion, and diversity at Georgetown as a member of the Racial Justice Committee for Change and as the director of the Medical Spanish Initiative. And with that, it is my pleasure to pass things over to Dr. Chris Albanese for his presentation today.
Thank you very much for that kind introduction. While I'm fumbling here to share, let's see, I'd like to thank VisualSonics for this opportunity. It's been a wonderful collaboration with them over many years, and we look forward to doing more stuff together. Let's see where I'm working from a laptop. It's not always my thing. Okay, I trust everybody can see this.
Yes.
Great. Okay, so, oops, did I go offline?
No, I still hear you.
Okay, great. So based on the introduction, I wanted to show at least some of the people who were involved in the work that we did. Of course, there's Olga and me on the left-hand side in front of our magnet. Yi-Chen Lee is the expert in MRI, and he's an assistant professor in the laboratory. And our expert, truly expert ultrasonographer is Kyle Karolowitz. They're here as well, but decided not to be on camera. This is the equipment we have. Of course, shout out to VisualSonics for the 3100. We have a relatively new Bruker 7T30 we use for a lot of workhorse imaging. Like most preclinical imaging labs, we have a IVUS system, X-ray system, and we've added now for fluoroscopy a human C-arm and a human ultrasound machine for intermediate to large size animals. As we said in the beginning, this is kind of how we are set up as a regional imaging center. We do everything from very basic science all the way through to clinical applications and back and forth using a variety of animal models to be that nexus between what's happening in the laboratory and what's happening in the clinic. And we have 75 or 80 papers in this field to date. Some of the things that we have done that we're wanted to highlight is that with our Center for Translational Imaging and the Physics and Chemistry departments here at Georgetown, we have patents out on T1 and T2 contrast agents for MRI, which attempt to get around some of the issues around gadolinium and its potential toxicity. This is a nanoparticle that uses manganese clusters that have iron oxide in them that give both enhanced T1 and enhanced T2 imaging. Anatomical imaging, we do quite a lot of that for any number of different diseases. This is a paper we published a few years ago looking at the anatomical structure of the mouse prostate. Of course, being a cancer center, many people are looking at cancer progression and metastases. And for those animal models that are not using things like an IVUS system that have luciferase activity, MRI is in many cases the only way you can actually see where tumors go. And this is an example of a lung tumor by MR. Metabolism, of course, is a very important topic for most any study, whether it's cancer or non-cancer. And we apply something called magnetic resonance spectroscopy to a variety of different animal models, both large and small animals. In this case, it using the spectra, the ppms of different biomolecules that exist in different tissues. We can look at changes in the tissue's metabolic profile as it goes through its disease state and whether or not certain drugs or interventions actually change that disease state. And again, this is all published data. I won't go into it, but it's another one of our workhorse technologies. A growing technology in the MR world is chemical exchange saturation transfer. This allows one to look at key metabolites such as glucose in this case, and its changes in glucose concentrations as tumors grow or regress following cancer treatment. And on the left-hand side, we also apply diffusion-weighted imaging, which looks at the diffusion of water through tissue, and that changes as the tissue goes from a normal to a neoplastic and back to potentially a normal tissue. MR is actually quite good for angiography. These are studies that we did with the Georgetown MedStar Research Institute to look at the changes in vasculature in aging animals. Again, I won't go into this, but using MRI as kind of our stable workhorse imaging machine, we're able to do a number of different things that a single machine like a CT or a PET machine can't do. And we continuously work with our investigators, both at Georgetown and at other institutions, to develop new technologies and new approaches to address key questions. So the reason I'm assuming most people are here are based on the fact that we have now published on using photoacoustic imaging to look at liver disease. Liver disease is a growing problem in the United States and in the world. It's now referred to as metabolic dysfunction associated steatotic liver disease, or MASLD. It used to be called NAFLD. And this occurs when there's an excess fat accumulation in the liver, and it's linked to conditions such as obesity, type 2 diabetes, and other things. And of course, there's also HCC that comes out of viral infections, which Kyle is an expert on working on. Anyway, so this graphic here shows that in the early stages of steatotic liver disease, it is reversible if you do intervention through dietary, GLP-1, other things. You can actually reverse some of this. However, once it progresses to a late stage, it starts becoming quite a fibrotic tissue. And fibrosis is basically the basis of many different diseases, including just aging. It can become a problem when it's unchecked. So once it becomes a steatotic fibrotic liver and goes through these severe stages of liver dysfunction and onto HCC, this is liver transplant or death stage. So it's a very key and critical issue when dealing with obese and diabetic people, but it also affects people across many different socioeconomic backgrounds, as well as people who have been affected with Hep B or Hep C. One of the problems that we're facing is there is a rising level of obesity in the American population and certainly worldwide that actually threatens the donor pool. One associates usually the issues with liver transplant with the patient being unwell. But of course, as people's livers start becoming less and less healthy, the potential for liver donation becomes less and less effective. And there's an obvious need to improve the health of both the transplant recipient, but also the donor themselves, as this MASLD and just metabolic liver disease increases in the general population. This was a study we published a few years ago with Alex Cromer, who's in the Transplant Institute here at Georgetown. And they are facing this exact issue that we showed on the previous figure, that while they do a tremendous number of liver transplants for both children and adults, the health of the adult liver population is decreasing. And this was a study that we performed with them to study the effects of N-acetylcysteine, which is basically, you can get it over the counter. People use it for head congestion. It loosens the mucus. But it also has metabolic effects systemically and in cells and tissues. In this case, N stands for normal diet, and HFD stands for high-fat diet, and we used MRI in this figure to look at the mean relative intensity of regions of interest within the livers of these animals. And in the right-hand side, you see the bar graph where the relative intensity of the livers in the normal diet changed only modestly with N-acetylcysteine. And the mean intensity value, this is again looking at adiposity based on the fact that fat is whitish in T1 weighted MR imaging. You could see that the level of adiposity in the high-fat diet fed mice increased and was brought back towards baseline with N-acetylcysteine, suggesting again that the livers may be more healthy. That's only one method of looking at liver health. This is dynamic contrast enhanced MRI to assess liver function. In this case, we're using Eovist, which is kindly provided by Bayer. It's taken up by the normal adipocytes and excreted through the bile ducts, and also to some degree through the kidney, but we're looking at the liver here. And the hypothesis was that if indeed the high-fat diet livers were less healthy than the normal diet, we would see changes in both the amount of contrast enhancement provided by Eovist over time and also the slope. So in the top, we see with the regions of interest that were selected in the baseline and over time, PI is post-injection of Eovist. And you can see that the tissues increase in their overall contrast, and we're able to select regions of interest, similar regions of interest in each of the livers, and did the analyses. So in panel B here, you can actually see the slopes. The blue is normal diet. Red is normal diet with, oops, I'm sorry. Red is normal diet with N-acetylcysteine. The high-fat diet is green, and you can see that both the amount of contrast enhancement and also the slope has changed, and those are to a very high degree mitigated by the addition of N-acetylcysteine. So this suggested to us that there was fat in the liver, that the livers were functioning improperly on the high-fat diet, and that a lot of this could be mitigated through the use of N-acetylcysteine. This is some ultrasound of a similar type of study that I did with one of our collaborators, looking at Western diet and intervention here. And this used ultrasonography to look at the echogenicity on the Western diet. And again, we can see on the right-hand side here that the echogenicity increased, suggesting that there were changes in the liver that were associated with a Western diet that could be mitigated using the intervention that we used, which was not NAC in this case. But again, there was an obvious need here, and these imaging approaches that we're using are very informative, but they don't actually get to the root of the cause of the changes in the pathology of the liver. And that's where we entered into this wonderful collaboration with Jiffin and the rest of the group at VisualSonics to use to continue to develop their photoacoustic imaging technology to assess, in this case, liver function in animal models of liver disease. And part of this was we had a day-long seminar here at Georgetown where local regional people came in to give presentations and listen to things that we were doing. And there was also a live demonstration of photoacoustic imaging using our 3100 system and the photoacoustic module that was on loan from VisualSonics. During that time period, we actually used a different animal model to start to assess whether or not photoacoustic imaging would be an effective and rapid way of looking at liver steatosis and fibrosis in a mouse model. And this is the paper. I'm sure many of you are here because of it. The references at the bottom and the list of authors and their institutions are also there. Anyway, so this is the animal model. Tally-ho mice are a spontaneously diabetic mouse model that we're collaborating with a number of other groups here at Georgetown to look at different components of diabetes and other things. And we know from previous studies with the group, including Blythe Shepherd, that the choline deficient diet in this animal model leads to a relatively rapid increase in changes in the liver that were associated with steatotic liver disease. We thought this would be a very good model system for us to follow using, typical kind of clinically relevant serum-based assays, as well as in vivo and ex vivo documentation of the amount of fibrosis and steatosis. So in this figure here, we're showing the effects of the choline deficient diet on the tally-ho mice. When the male mice went on this diet, they kind of lost some weight, but it came back to basically baseline after 6 or 8 weeks. The females did not lose any weight. Interestingly, but not interestingly, we expected this. The liver weights increased statistically significantly in both the male and female mice, and the kidney weights actually decreased. And again, ALT activity is something that is clinically relevant. That's what's used to assess liver function in humans. And again, both the males and females had statistically significant increases in ALT activity, again, a hallmark of liver disease. Falling back on T1 imaging, which we showed in the previous study, we found that the relaxivity time of the T1 relaxivity time in the male mice had a trend upwards, but did not reach statistical significance, probably because of the number of mice we studied, but how the female mice were statistically significant increases in the T1 relaxation time and a statistically significant decrease in the dynamic contrast enhanced EOVIS data. All this were suggestive, of course, of kidney disease. Another trick of MRI is T2 imaging, and we applied T2 imaging to both the male and female mice on the control and CDD diets, and we showed that the T2 relaxation times were statistically significantly increased in both the male and female models on the choline deficient diet. And again, this is it's a general indication of fibrosis and/or inflammation. It did show that there was kidney disease going on, but again, it didn't get down to the root cause. Was it the inflammation? Was it fibrosis? Was it a combination of the two? Oh, did I say kidney? Yeah. Liver, excuse me. So here's where we get into the meat. These were studies that were done here at Georgetown. Lori Rich and Kyle Karolowitz did the ultrasound and photoacoustic imaging on the animal model I just showed you, and the animal models I just showed you. And Jithan and the group back at Fuji, VisualSonics, did the chromophore abundance mapping and then deconvoluted it using their SPACS photoacoustic unmixing technology. Questions will have to go to Jithan on that, but that's what was used. In this figure, we're showing the deoxyhemoglobin imaging. So again, this is a method of looking at the changes in oxy to deoxy hemoglobin levels in the livers. And what we find here is that in both male and female and male mice, especially compared to the control diets, there were statistically significant increases in the deoxyhemoglobin levels in the livers of these mice. The next figure is doing the same type of analysis, looking at lipid content, which is in the orange pixels here. So again, in both the female and male mice, when put on the choline deficient diets, there were statistically significant increases in the image analysis of the liver content of lipids. Finally, as far as the photoacoustic imaging, we applied that, of course, to fibrosis. This took a while to get correct. It was a number of different attempts to properly train the photoacoustic SPACS method to pick up the collagen that was necessary for this type of imaging. And you can see here again that in both the female and male mice on the choline deficient diet, there were huge changes in the amount of fibrosis that was imaged by photoacoustic imaging and ultrasound. And my final data slide is, again, the ground truth pathology. And on the left-hand side, we're showing picroserous red staining under polarized light of male and female mice on both the control diets and the choline deficient diets. This is a very nice quantitative measure of fibrosis that is now quite often used. And our chief pathologist at Johns Hopkins did all the reads. So we're quite confident in these data. And you can see that in both cases, the choline deficient diet causes significant increases in the collagen content in the livers. Equally so, we see steatosis in both the male and female mice on the choline deficient diets, statistically significant with both males and females. So the conclusions are fairly obvious, but important. The PAI framework with SPACS presents an exciting enhancement in the medical imaging with a potential to enhance diagnostic and therapeutic monitoring. The limitations of our study, of course, are that the depths of penetration are somewhat minimal at this point and best used in a preclinical study for at least liver disease, although it probably could get to the upper level of a human liver, but not into the depths that we showed in the mouse model. And ongoing research is obviously different light delivery systems, fiber optics, and other ways to overcome the limitations that will hold back, at least for some time, the ability for us to do similar types of studies on human patients before and after liver transplant. So that's basically it. I'd like to acknowledge all the people that were part of this. All the imaging was carried out here at Georgetown. We had buy-in from groups in Milan, Italy. Again, the Laser X module was on loan from VisualSonics, and that was obviously a great seminar meeting, and the follow-up studies that we did were wonderful, and we appreciate it very much. Again, Bayer provided the EOVIST, and we were supported by an S10 grant for the MRI, a P30 grant, and other grants that covered some of the other work that we did here. And any contacts necessary, you can contact either Olga or me, and our email addresses are at the bottom of this slide. Okay, that's it. I guess I will stop sharing.
Great. Thank you so much, Dr. Albanese. That was great. So clinically relevant. It's nice to see that kind of bench to bedside to bench style of going through your research program. That's excellent. So at this point, I'd love to introduce or encourage people, I should say, to put in your questions in the Q&A button in the panel. There's a couple that have come in through chat and through Q&A. So we can start with that. And please, by all means, filter some more questions in as they come up or come to mind. So the first question is, how were MRI image intensities normalized across multiple scans and days?
I'll let Dr. Rodriguez answer that.
When we talk, whichever way we do it, we usually normalize against a tissue or the background. So for instance, with the EOVIS is a contrast agent that is taken up only by the liver and only by the kidneys. The rest of the tissues do not take it up. So in that case, for instance, we normalize against the contrast of a muscle, for instance. So it depends on what specifically we're doing. So when it comes to contrast agents, it depends on the type of contrast agent. When we do T1, T2 imaging, there's no need for that because the normalization is intrinsic in the protocol.
Great, thank you. So the next question, compliment, great work, very interesting. It's nice to see the in vivo lipid and collagen detection. The question was, did you also look into oxygen saturation differences? And I'll add to that, I know you shared some differences in deoxyhemo in the diseased livers. Can you elaborate on why you think that you saw the decrease in deoxyhemo?
Yeah, it's a good question. We don't have an exact answer for that. It could be perfusion differences. It could be metabolic issues within the tissue itself. Yeah, there are a number of things that impact both oxy and deoxy levels. So it's hard to know. It's hard to know. We would have to do additional studies to figure out whether it's blood flow, any metabolic changes within the tissue. There's definitely plenty of those.
Inflammation.
Yeah, inflammation is another component that is part of the liver injury. So there are multiple things that could change that. We don't have a specific answer though.
Okay, great. So a couple questions about differences between male and female mice and how they reacted in the study and what you saw. Noticed, was it expected that the female mice on the CD diet did not lose weight and yet still exhibited the signs of pathology similar to the male?
Yeah, again, while we don't have an answer for that, we have seen these things in other experimental experiments that we're doing. The males do seem to respond differently initially to the CDD diets, but they do moderate that to some degree. It doesn't look like it's just simply food intake. It could be a metabolic change. As far as some of the imaging outputs, again, I think there's only one or two instances where the males and females were so different that one was statistically significant and the other one wasn't, and that's probably just because we only used 4 or 5 mice per study group. And we would anticipate that, given that the ground truth pathology was showing us that both animal models got both steatosis and fibrosis, that the inability to reach statistical significance in some of the imaging approaches was simply just a matter of, the variability in between animals causing the variance to be high enough where the p-values fell below 0.05. And again, males and females aren't the same. So I guess it may not be necessarily surprised that one sex, I guess they're called, it would respond differently to a dietary change or, temperature changes. All those types of things are different between males and females. Hormonal, so we don't have an answer why the males lost weight. It didn't seem to be simply just not eating the food, but we don't have an answer.
Okay, thank you. On looking at other organs in these models, did you see any cardiac phenotypes in your marine liver disease models? Or if you didn't look at that, would you expect there to be a cardiac phenotype?
Well, we have looked in other model systems at the impact of Western diets on the cardiovascular system, and there are impacts. What we would like to do in the future, of course, because we only had a certain amount of time to do this, again, and I think I misstated that we looked at this past study was in the kidneys. We were very interested in what's happening in the kidneys. It's a major site of fibrosis, being clinically relevant disease outcomes. The cardiovascular system, we do have a number of other studies going on, but unfortunately they're not taking advantage of the photoacoustic capabilities because we don't have a module here. So yes, we would expect there to be some changes in the cardiovascular system. And we have seen changes in the cardiovascular system with certain high-fat diets and certain animal models. but we don't have photoacoustic data on that. We have ultrasound, we have MR, angiography, and we have, of course, pathology.
That's great. And on the photoacoustic note, some of our audience members here might be a little less familiar with what photoacoustics is. Do you mind giving a brief overview of that technology?
In its simplest form, and Jithan is online and he can pop in at any moment if he wants, it's basically under ultrasound guided imaging, a pulse of laser light is shown into an area of interest, and that light interacts with different chromophores within different molecules. And those signals are read back into the ultrasound wand, and then translated into, I'm sure through Fourier transform, into data that Jithan and VisualSonics can then deconvolute to assign a specific frequency, so that the laser light is actually pulsed over a frequency range. So similar to what I showed in the MR spectroscopy, different molecules have different signatures that one can pick up and then turn into visible data. And that's the basis of this, that fat has a different signal output from the photoacoustic input than fibrosis does, than deoxyhemoglobin does, than oxyhemoglobin does. And then that deconvolution that Jithan does allows us to visualize that and overlay it onto the ultrasound image to show regions of interest that have changes in those signals.
Yeah, and I'll just add to that as well. So from a product perspective, we have our ultrasound system, and Dr. Albanese is using our Vivo 3100, which is the high frequency ultrasound. To do photoacoustics, you combine that or pair that with a near infrared laser that you can deliver the light kind of alongside the ultrasound transducer. And then the basics of photoacoustic imaging is light in and sound out. So the light causes thermoelastic expansion, like Dr. Albanese is saying, at different wavelengths, depending on which component you're illuminating. And then you're able to spectrally unmix different characteristics in the body and co-register those images of that photoacoustic data that's looking at exogenous or endogenous markers in the body, along with the anatomical data you get from the high frequency ultrasound.
Yep.
Okay, another question here. Do you see any adverse effects, or can you speak to maybe some of the disadvantages of using MRI in mice?
No, there are no disadvantages except time and money. To using MRI, it's the do-all, find-all piece of equipment in this laboratory and in every hospital in the world, because it can be used to look at everything, as we showed, from contrast agents all the way through to, we're using it to look at changes in brain structure and function associated with aging. There's almost nothing that MRI can't do. Some of it doesn't do well is to look at things that are very, very low abundance or super small. So that's where things like CT and PET come in for specific uses. But there are no adverse effects at MRI. For those who don't know, it does not produce radiation. It's just a big magnet. And the contrast agents that we use, gadolinium-based, are used. It's $1 billion industry. So every hospital uses it. So that's part of the translation that we use, clinically relevant approaches for preclinical models that can then go hopefully back to the clinical applications for advancing disease treatment or identification, with some of the stuff that Kyle's doing with hepatitis infections. They're looking at using new devices for basically ablating the tumors using larger animals, and that's where the larger animal ultrasound machine and the Vivo system is used in combination to look at different depths. So yeah, no, all these devices are good, but MR is the most flexible.
Versatile.
And versatile.
Okay, great. And looking at that the methodologies you've used here. Someone's asking, it's a bit off indication, but in endometriosis work, if you're familiar or can extrapolate to how the technologies you've used here could potentially be beneficial in doing endometriosis research.
I'll let Olga, I mean, probably the fibrosis could work there, but
Yeah, endometriosis is very challenging, you know, and when endometrial tissue is going to be all over the abdominal cavity, and already the intestinal tract is complicated in terms of imaging and in terms of being there. So, like Dr. Albanese was saying, perhaps identifying fibrosis, but still, I think it's that would be a very a challenging disorder to study still through any type of imaging at this moment.
yeah, I agree.
Yeah.
It's a toughie. Yeah, there's a lot of stuff going on in the abdomen and
Yeah.
you know, MRI will help, but yeah.
Yeah, and there have been some studies using endometriosis models in preclinical studies that have used ultrasound to visualize those lesions. So we can definitely provide some more information on that front as far as what's been published so far. Not necessarily looking at fibrosis specifically, but using ultrasound anyways.
Yep.
So another question here, did you ever look at the brain of these animals, or would you ever expect there to be any changes in the neurobiology?
Oh, of a diabetic mouse on a high-fat diet?
Yeah.
Probably. We have not done that. That's, you know,
Oh,
we have.
DB mouse.
Oh, right. Yeah, we did look at the diabetic mouse, the DB mice on high-fat diet. We did see changes by spectroscopy.
In both cortex.
In the cortex and the hippocampus. Thank you. I forgot about that. Yes. So the answer is yes. There are changes. The spectra gives us that information. I don't think we did the voxel-based morphometry, but another trick of MRI is that you can do something called voxel-based morphometry. We actually published this with some of our colleagues here at Georgetown in the neuroscience department, looking at the impact of the ApoE3 and ApoE4 allele, which are genetic predictors of Alzheimer's disease. They actually also can, especially ApoE4, promote a sensitivity to chemotherapy in older women who are getting breast cancer treatment. And we found that the ApoE4 allele actually by voxel-based morphometry, which is a high dimension, high resolution 3D image mapping of the brain, we found areas in these knock-in mice that had either the human ApoE3 allele or the human ApoE4 allele. We found that there are areas just by virtue of the one amino acid change between those two genes, changes in the the structure of the hippocampus and the cortex, and those changes were exacerbated by treatment with doxorubicin, which is a first-line therapy for breast cancer. So, and we're about to publish another paper along those lines. So yes, indeed, you know, we do look at the brain a lot. We have done it in the DB mice. We did not do it on this model that was just discussed using the tally-ho mice.
Okay, great, interesting.
Long-winded answer, but there you go.
Yes, no problem. Do you have any insights into how liver disease impacts metabolism? This researcher is exploring the use of phosphorus 31 MRS for liver imaging.
Yeah. Oh, wow. It's everywhere. Yeah, I mean, I think probably the data that are coming out of the GLP-1, you know, studies where people are, you know, losing weight rapidly and their metabolism is changing, and it's helping both cognitive skills as well as, you know, the health of the animal, the human in this case. It's everywhere. It's insulin. It's, you know, the adipokines, adipoq. I don't even know where to begin. There's so many changes that take place when somebody is either obese or then starts losing weight and becomes less obese that, yeah, I don't even know where to begin on that. There's many very recent, you know, reviews in Nature, Science, and other things that go into some of this, but yeah.
The big can of worms, right? It's a burgeoning
Big can of
field.
worms. Yeah.
Absolutely. Great. Another question here. First, a compliment. Thanks for the presentation. Have you conducted any studies looking at tumor growth and characteristics like imaging or pathology in steatotic or fibrotic liver versus otherwise normal livers? Any insights you might be able to share from such studies?
We do have a study with one of our colleagues who has genetically engineered mice that are predisposed, and it's not published, so I can't go into it. But the answer is yes, we are looking into that. And it might appear that having a leaner liver is better for tumor growth, but we don't know that yet.
Okay, interesting.
Good strains, in fact.
Yeah.
I know, yeah, obese and lean.
Yeah. Yeah. But yeah, not published yet. But yes,
All right.
we are interested in that for sure.
Interesting. Yeah. So keep an eye on Dr. Albanese's publications coming out, and we'll have the answer soon, hopefully. I noticed a couple questions about software. So I'll just kind of address that in that all the software for analysis on the ultrasound side is performed using our Vivo Lab analysis software with a beta of our spectral unmixing algorithm that we've been testing it with certain sites. So we're more than happy to elaborate on that, and we can contact you directly if you're interested about that. Question here. Have you done any research or data looking at pancreatic cancers or looking at these methodologies in pancreatic cancer models?
Yes, we are doing that. Again, we don't have the photoacoustic module. So whatever we do is based on ultrasound and MRI. But yes, we have done both and are doing both. Kyle is using the VisualSonics system to do pancreatic injections. Robotically assisted pancreatic injections, and then we apply both ultrasound and MRI to look at relatively fast, simple assessment of tumor establishment, and then the MRI gives us everything from the anatomical image with no depth issues whatsoever, as well as metabolic information on the tumors.
Perfect. All right. So I think we'll finish out with one last question here, but if as you're answering this one, anyone has any additional questions, please type them in the Q&A panel. We will direct your questions and have them answered after the webinar. I know we've peppered Dr. Albanese and group there with many, many questions. So here's the last one here. In your preclinical MRI T1, T2 imaging studies, did you encounter significant motion artifacts due to cardiac or respiratory activity?
No, not in this case. We do have to deal with that certainly, but we do gating, which of course increases time, but also decreases motion. There are certain, protocols that we do have to deal a little bit more with the animal motion. And if we don't gate, then we don't get such a great outcome. But we do the respiratory gating through Bruker. It's all part of the system. So we can either turn it on or turn it off. These were done with it on.
That's great. All right. So at this point, I'd like to thank Dr. Rodriguez and Dr. Albanese and Kyle as well so much for the very interesting work. You have a great group there and lots of really interesting research going on. For everyone joining us today, thank you for coming and all your interactive questions. It's always great to see. At this point, I'd like to encourage everyone to please stay connected with us. We love speaking with you about your research. You can contact us on our website through our customer resource portal. You can submit any questions you might have if you're wondering if something might be possible with our equipment or if there's been publications. We can definitely help you out with that. And there's a whole publication library of all publications that have come out over the years, over probably close to 7,000 or more publications using our technology in different animal models. Happy to share those citations with you. We do have an exciting webinar program of past webinars that have been quite recent and then future webinars. So please check out our website in the webinar section to register for any future webinars. We would love to see you. We're active on all of our social media channels, so please follow us. And we also visit several scientific conferences, and we'd love to see you in person and come visit our booth. So please check on our website to find out when we will be at the next conference you're attending. Finally, on the note of conferences, the liver meeting on the topic of our webinar today is coming up very soon. I'll be flying out to Washington on Thursday, actually. So I'd love to have anyone come visit me and the rest of the group at VisualSonics group at our booth at the liver meeting in Washington in the next week. So with that, I'd like to thank everyone so much for joining us. Thank the speakers again, and I hope everyone has a lovely day.